Yeah. Uh, Very pleased to welcome Dr. Shachi Mittal. She is assistant professor in the chemical engineering department at Udub. She received her PhD from bioengineering at UIUC in 2019, and she's also graduate of the IIT Indian Institute of Technology in India. And she has a lot of expertise. Like, she comes from a chemical engineering background, but she has expertise in cancer biology, pathology, and AI. So she's really interdisciplinary, and so and she has a lot of publications and awards as well. So I think it's a great opportunity to learn from Shachi's experience and get to know more about hands-on in cancer detection. Shachi,
yeah, thank you, Karthik, for your introduction and for having me here. So hi, everyone. Today I'll be talking about my work on AI-assisted cancer diagnosis. Okay, let's see if this is. So let me just give you a brief primer of how cancer diagnosis happens in clinics, and then talk about how engineering and AI can interface with this pipeline. So when a patient comes in with a suspicious lesion or a suspicious area that they think is cancer, that part of tissue is taken out from that patient. So tissue is a 3D blob, right? So then that 3D blob is sliced, and each of those slices are placed on glass slides. So each of these slides are coming from the same patient. Then they are stained by a variety of antigen-antibody interactions to highlight specific molecular markers. And then they are imaged under a microscope, which are visualized by a pathologist or a clinician who looks at cancer diagnosis. The biggest problem with this approach is because it's an observer-dependent diagnosis, it's difficult to standardize from one clinic to the other. There can be measurement errors that are coming not only from staining the sample, but also interpreting the images after you've stained the sample. And there is limited field of view and limited use of the microenvironment of the sample, which I'm going to talk about in the subsequent slides. So just so that the talk doesn't seem like jargons to you, I'm going to give you a 1-minute primer on what breast pathology is. So throughout the talk, the application in cancer that I'm going to be talking about is breast cancer. So how do clinicians actually diagnose breast cancer? So when there are cells that are growing in one layer and one cell acquires a mutation, and these cells start proliferating, which means that they start growing on top of each other, that is called as hyperplasia, or in other words, hypergrowth, okay? Then when there are multiple types of mutations, like we have the orange cell here and the blue cell here, that's when we call as dysplasia. Up until this point, there is no cancer, but it just puts the patient at a higher risk of developing cancer. For example, dysplasia is about 4 to 5-fold higher risk of developing cancer. But when we have the carcinogenic mutations, which are the red cells, but they are contained within this gray membrane, that is called as in situ cancer, or the cancer that is inside. And that is called as stage 0 cancer. But when these cells break the membrane apart and spread to the rest of the body through the lymphatic system, that is called as invasive cancer, which means that it has invaded the space. Okay. Now talking about the microenvironment, when these changes are happening in these blue cells, which are these epithelial cells, there are a lot of changes that are happening in the collagen or the fibers that are surrounding these cells. And that is called as the microenvironment. Environment is the environment around the cancer cells, micro because these are all micron scale cellular entities. Okay. So how this translates to patient images. So here I'm showing you 2 images from different patients, and the cells are in purple and they're very well organized only in one layer. So we know that these are normal cells, and these fibers around these cells is called as normal collagen. But when these cells start growing on top of each other and are ugly looking cells, which are also in clinical language referred to as atypia or atypical, which is dysplasia, and they are still contained within the membrane, that's where I know that these are stage 0 cells. And you see that the collagen around these cells immediately changes its behavior. So the collagen is no longer the pink dense fiber, but it is developing a lot of different nuclei and is also developing different colorations. And that is referred to as desmoplasia, which is a typical microenvironment change in breast cancer. And similarly here, the cancer cells have invaded the entire space, which is advanced cancer, and we have desmoplasia. So now you know about the breast pathology that I'm going to be referring to in the talk. So in my initial stages of research, I started looking at novel bioimaging technologies, which are not being used in clinics today, to get more information about the patients. So the first imaging technology that I worked with was infrared spectroscopic imaging, where all the chemical bonds vibrate at certain frequencies. And those frequencies lie in the infrared region of the light. So when we throw light, which is in the infrared spectrum, what happens is that different chemical bonds absorb those lights differently. So a carbon-oxygen bond is going to absorb that light differently versus a hydrogen-oxygen bond. And based on this, we assume that if we are able to throw light of thousands of frequencies, acquire it over millions of pixels or data points, we should be able to get profiles that are going to be different across lipids, biomolecules, and other entities which are different from normal cells and cancer cells. So here we have the tissue. We take out areas of interest and we create something called as a microarray, where we can, in a small sample area, we can sample heterogeneity from different patients. We throw infrared light of thousands of frequencies to get an image like this, which is obtained at one frequency. So this is just an absorption image. So the points that are white are basically areas that absorb that particular light a lot. And the areas that are black, those are the ones that did not absorb that light as much. And this is the spectra that we get at every single pixel. So there are about 1 million pixels in this array. And we are able to get information about lipids, proteins, nucleic acids, and carbohydrates, which changes when a cell goes from normal to cancer. But the problem is, you can imagine, these are thousands of frequencies over 1 million pixels. You cannot recognize these patterns by naked eye. You have to approach pattern recognition methods to be able to find out profiles that are specific of cancer and normal cells. Yes.
Yeah, are these frequencies predetermined, or it's the highest one spectrum?
So these frequencies, once we fix the instrument, which is operating in the mid-infrared region and is a broadband source of light, this is fixed because it's kind of like a light bulb. We cannot control what exact frequency it's throwing. It's going to throw the frequency in the entire range. But later in the talk, I'm going to talk about how we shifted this to a discrete frequency instrument where we can specifically choose the wavelength that we are throwing at the sample. So I think you're all aware of this. So we resorted to machine learning to extract knowledge from this data, which is about many terabytes across just one tissue microarray. And we can do unsupervised learning, which is just finding trends in the data, finding new patterns. But that's not something that I'm going to talk about in the talk today much. We basically shifted to supervised modeling, where we first identified the specific features in our data that are indicative of discrimination across different classes. So the data looks like a 3D cube, where in the XY space, we have the spatial coordinate of the sample. And in the Z space, we have the absorbance at each frequency. So the dimensionality of the Z space is equal to the number of frequencies that were thrown at the sample. So if I had about 1,000 frequencies thrown at the sample, I have 1,000 dimensions in the Z space, which can be very large for building any efficient machine learning or deep learning algorithm downstream. So the first step is selection of features, which we call as dimensionality reduction. And we identify certain subset of features that are giving us the maximum separation across classes. Then we collaborate with clinicians who are able to give us an exact label for certain pixels on this image that are either cancerous, which is referred to as malignant, or non-cancerous, collagen, and other cellular components. And initially, we worked with traditional machine learning approaches. So we used a random forest classifier. And I'm not going to go into the details, but if somebody has questions, we can go over that in the questions. And then for each pixel on the image, we could get an assignment of what it belongs to. And then we compare it with the stained image that is the current gold standard in diagnosing cancer to calculate the accuracy of the model. When we saw this, I was curious whether, so when we talk about interpretable model development, right? I wanted to see if the features that I'm picking up are capturing a chemistry that is similar to what clinicians look at when they stain the sample. So what I did was that I repurposed some of these features, applied a different color map to get something which I call as digitally stained image or a stainless stained image, which is very similar to the H&E image, which tells me that there is a similar chemistry that is present in my feature set that I'm trying to capture using machine learning.
So,
yeah.
Although we are sampling across multiple frequencies, the annotation is like looking at all of that and just assigning a particular spot as being malignant or not.
Yeah, so the annotation has nothing to do with frequencies. It is only looking at the spatial architecture where it's only looking at the XY.
Okay.
And for every XY, we have basically the Z space. So after we trained the model, what we could get was an assignment for every single pixel of this array, which is 1 million assignments. And throughout my talk, wherever you see a color that is closer to red, that is something that I'm unhappy about, which is closer to Cancer. And whenever you see a color that is closer to green, that is something that we're happy about. Okay. So there is a lot of information here. These are 100 patients of breast cancer. I'll zoom in on 4 different patients. So the first is malignant, which is the advanced cancer. And remember, it was the invasive cancer that was spreading everywhere. And the model is able to identify all the cells as belonging to malignant. On the second and the fourth case, which is normal hyperplasia, we also went through, which is normal, but a higher risk of developing cancer. And the model still finds that these cells are all benign, which means that they're normal. Let's look at patient number 3 or patient D2, which says atypia, which is the category of dysplasia, which was 4 to 5-fold higher risk of developing cancer, but not cancer. And what the model figured out was that there is actually a mixoid population of cancer cells and normal cells, which the clinicians wouldn't be able to see. And why that is happening is because when we look at infrared spectroscopy, we are trying to detect a biochemical change. Whereas in clinics, we are trying to detect a morphological change. And a biochemical change is always going to precede a morphological change. So this kind of gave us cues that this can also be used as stratifying or detecting patterns early on. And for developing any diagnostic test, whether it's imaging-based, whether it's marker-based, to be actually approved to be used in clinics, you need to do the sensitivity and specificity analysis. Sensitivity is how sensitive my model is to identify something that exists. Specificity is how specific my model is in distinguish between the various scenarios that can be present on that location. Okay. And this curve is called as ROC curve. And a measure of accuracy is the area under the curve, which is called as the AUC value across this curve. An AUC value of 1 is a perfect diagnostic test. So we want the value as close to 1 as possible. So for all our classes, the values were above 0.97 across these different categories. And when we look at just the first or the top 7 features, the accuracies were still more than 85 or 86% across these different diagnostic categories. So here we can see that there are different variations, but we took the same data, applied the same model, but now our classes are slightly different, where we wanted to separate these cells of different stages of tumor development. And looking at these patients now, if you look at again patient number 3, the model is able to identify that dysplasia or atypical hyperplasia very accurately, again with an AUC value of more than 98% across these diagnostic categories. Now, this is all good, but the problem with this is for me to image each circle here, which is 1 patient, it took me 3.5 hours to just image the patient, and then you project the classifier because this is a high definition imaging. So we knew that 3.5 hours is not something that clinicians are going to be happy about in terms of translating a technology from lab to an actual clinic. So we figured that when we use just the 7 features, the drop in accuracy is not that much. So how about we move to discrete frequencies where we don't have to throw all these thousands of frequencies because anyways, there is redundant features there. So we moved from translating and expediting our data acquisition, where we can not only now look at tissue microarrays, but also look at large specimens. So microarrays is typically what is used in labs or sometimes in clinic to develop something, to study something, to do research. But when a patient comes in for diagnosis and treatment, this is the area that is taken out from just one patient. So this is the millimeter scale here, and this is the millimeter scale here, right? So if this circle takes me 3.5 hours, it's going to take me months to just acquire this image. So we move to discrete frequency measurements, where now we have the same microarray, but we only threw 12 frequencies instead of 1,506 frequencies, to be precise, to develop the model. And here again, we were able to separate atypical cells from the collagen, compare it with the gold standard, and also look at the changes in the microenvironment. And obviously now, because we're using a subset of the features, there is going to be a drop in the accuracy. So there is about a 10% drop in all classes, but it is still more than what the concordance is across some of these categories in the clinics today. So we were basically able to achieve 50-fold increase in data acquisition speed with respect to this. And then we extended this to a collaboration with a clinical lab, and we got the specimens that they typically would receive for patient diagnosis, where the clinician told me everything that is circumscribed by a blue dot is cancer, and anything that is circumscribed by a green dot is normal. And here I'm showing you a representative infrared image that was taken at 1 frequency, which gives information about proteins. Amides bonds are present in proteins, okay? And this is the digital image on the bottom panel from the model that we had previously developed. And it's able to identify all the cells here in red as malignant, which if you look at this zoom, it should be cancer because it's circumscribed by blue dots. So we are able to accurately sample, and just within 3 hours, we could do all these 4 specimens with computation, with acquisition and data processing, which is much more than what we were able to achieve there. So here the 2 millimeter scale is just as much. This is much larger than a tissue microarray.
Question. The annotation itself doesn't change between the subsample and the other. So my question is, like, how exactly are we getting that patient there? Is it based on
Yeah, so the annotation, so clinicians don't understand infrared image, right? Because that's not something that they're used to looking at in a clinical lab. What they understand is this image. So the annotations that we get is always on the H&E image, which is our gold standard. We have to transfer those annotations to the infrared image, which is the adjacent tissue that was imaged, right? And we are transferring it in the XY space. So what happens in the Z doesn't really change the annotation. Whether I'm throwing all the frequencies in the Z, whether I'm throwing 1 or 2 frequencies, that wouldn't change my annotation. But I'm going to talk more about the challenges of annotations as well to scale up data science initiatives across imaging as well as clinical translation. So once we developed models in this area, I was curious whether some of these models can be translated to different types of imaging instruments, right? So at that time, a postdoc was working on developing a hybrid instrument that combines the power of infrared and visible microscopy. I'm not going to go into the details of the instrument, but if you just look at panel E, with again 90% accuracy using the similar model structure and architecture, we were able to separate cancer from no cancer on a completely different imaging instrument. But again, the problem is when you try to scale this up across different patients, the problem is of the ground truth data, because we have to actually hand transfer the annotations from the H&E to the infrared. And after I did that for about 500 patients, I was like, no, I need to develop an algorithm out of it and not having to do that by myself. So here what we did was we developed an algorithm which is freely available to the community, where you can transfer annotations from the H&E image to your microscopy of choice. In this case, we used infrared microscopy, and we use image registration and also cues from a supervised algorithm. So how it works is that it has a graphic user interface where anybody would select 3 data points that are similar on the H&E and on the IR, and it's going to register based on those 3 data points. Once it registers, there can be some errors which it then corrects for by this simple classifier that we have provided, because a cancer cannot lie on collagen, or collagen cannot lie on the green region. So we use those rules to correct for any errors in the registration, and it's able to transfer the annotations digitally. So this is what the interface looks like, where if for somebody who is not well-versed in data science, they can also use this platform to start generating ground truths data. You select the 3 points and it registers and transfers the annotations. So here on the top panel are handcrafted or transferred annotations by me. And on the bottom panel is the automated annotations that were transferred by the algorithm directly from the images that we got from the clinician. And once we did this, I wanted to explore a little bit more into the feature selection area, where we wanted to see if something that is stage 0 cell here on a clinical image, is it really stage 0 on infrared? Because remember when you saw myxoid signature on atypical hyperplasia, we said that something that was morphologically belonging to one class may be belonging to something else from a biochemical standpoint. So what I did was we first applied a supervised algorithm. We extracted just the epithelial compartment, which is where cancer originates. And we applied a clustering technology or clustering method to transfer or to see different patterns in this epithelial class. And what we immediately could see was the clinician told us that on the left-hand side of this image, we have stage 0 cells, and all the cells on the right-hand side are basically invasive or advanced stage disease. But there is some mixed cells that it was able to find out. And if we increase the number of clusters, again, we see that myxoid group. So we can use this to extract pixels that are actually pure in the infrared domain, which would really help us train these supervised models even with higher accuracy than having to just rely on morphology. So here we kind of showed how using supervised and unsupervised learning combined together can inform our models in a much better way. So the impact of this work was we were able to develop something that is quantitative, that can give us information about the cancer cells as well as the changes in the microenvironment, and is easy to standardize. We could get a 50-fold reduction in time, and this approach is compatible across different imaging instruments. But again, the problem is that infrared imaging technology is not a clinically approved technology. So there is going to be still a lot of time in taking these technologies to actual human impact. So then we wanted to shift and translate some of these digital approaches directly on clinical images. So that is where we started working directly with H&E images and identified textural and spatial features in diagnostic profiling. So here we tried to follow a pipeline that is similar to what a clinician would do, where we extracted regions of interest from these H&E images at different levels of zooms, applied a convolutional neural network to separate our epithelium, collagen, and other cells, and then clustered each of these components separately to identify various heterogeneous cell types that are present in these compartments. So here I'm just showing you the training data. The tiles were obtained of various sizes to get view at different zoom levels, right? Because if you see a nuclei or a cell from very far away, the edges that the convolutional neural network is going to extract is going to be very different than when you look at a nuclei from close by. So here on the bottom panel, we are showing the H&E image. And on the top panel is the digital image from the deep learning algorithm, where we could sample out the epithelial cells where cancer happens, the collagen, and other cells like blood or necrosis or secretions or dead tissue. And then we clustered the epithelial cells as well as the stroma compartment and looked at the inertia, which is the dispersiveness of each cluster. And what we noticed, and this was very surprising, we were not hoping to see this, but what we noticed is the inertia actually increased as we progressed from normal to a higher stage of disease. And the inertia and the collagen actually decreased as we progressed from normal to a higher stage of disease. And our hypothesis is when cancer develops, there are so many heterogeneous signaling that is happening in the cells that there is a disruption of your physiological architecture. And that is increasing your dispersion in the epithelial domain. Whereas in the collagen, which is the microenvironment, there are a lot of coagulation signaling that is happening, which is basically decreasing your inertia in the collagen space. So just by looking at the inertia in the epithelium and the collagen across this dotted decision boundary, across 100 patients, we were able to separate cancer patients by 90% accuracy just by these two features, which is the inertia in the epithelium and the inertia in the collagen. And you can see that we also see a separation of the normal cells, which are predominantly in the fourth quadrant, and the dysplasia cells, which are spread across in the quadrant, where some of these high-risk patients would actually be closer to a cancer signature than to a normal signature. And something like this can be very helpful for clinicians because it can tell which patients require more surveillance or aggressive treatment versus which patients can just go on with active surveillance without more aggressive treatment options.
So this can have this result in the extreme, right? Like
Yeah.
You're not able to differentiate dysplasia and hyperplasia,
Yes.
But they will fall apart because of inertia.
Yes. Yeah.
The places where you have less data,
Yeah, places where we had less data, and also something like whenever we build deep learning, the biggest problems that clinicians have is what is the quantitative variable that you're looking at, right? So here we actually kind of gave them that map that what we're looking at is the dispersion within your cells and the dispersion in the microenvironment. And what we are saying is that the dispersion in your cells where the cancer is originating is increasing, and the dispersion in your microenvironment is actually decreasing. Something that they can also relate to from a biological standpoint. So here we basically showed a digital textural feature analysis, which is simple and interpretable, and it gives us a multidimensional view of the disease, and it can be integrated with the scanners that currently exist in clinical labs to give them real-time patient analysis. But again, this still doesn't give us an exact view of early stage disease. And why do we want to detect something early on is this was a recent study where they gave about 250 cases to 200 pathologists all across the world. And they looked at the concordance, which is the percentage agreement between the clinicians. Invasive is advanced stage disease. The concordance is good, 96%. Benign, which is normal, the concordance is 87%, which is bad, which means that in 13% cases, they did not agree. Early stage disease, it's again bad, 84%, which means in 16% cases, they did not agree. Atypical, which is the dysplasia, which was 4 to 5 higher fold risk, the percentage agreement is worse than flipping a coin, which is 48%, which really showed that we need intervention or assistive technologies that can help clinicians with early stage disease segmentation. Whereas if you look at the patient survival, when you look at advanced stage disease, the 5-year survival rate is really small. So in terms of making an impact, you really need to operate in this region. Okay. So then I basically ended up developing a deep learning algorithm that specifically identifies early stage disease, which is ductal carcinoma in situ. In situ was inside the membrane. Okay. So here again, we try to mimic what a pathologist would do. So what a pathologist does, they basically take a slide, they put it under a microscope, they would zoom in, zoom out, look through the eyepiece, and try to look at the structure of the cells. So we try to do something similar where we extracted images at different levels of zooms, which is an automated pipeline that we created, where it's going to sample different areas at different sizes, and then it passes through a convolutional neural network to separate early stage cancer from the benign cells. We used 33 surgical specimens, which were the large specimens, giving us about 20,000 images to train, about 9,000 images to calibrate our hyperparameters or fine tune our hyperparameters, and another 9,000 coming from 11 totally different patients to externally validate our algorithm.
Can you say that the z-axis is the zoom? It's the 3D clock. Like in infrared, we have the frequency.
Yeah.
Can you say zoom is like?
So these are basically 2D images. There is no 3D in this.
Zoom is giving a 3D.
But we are not, the input is not as a 3D image. The input is still like in deep learning, we would have either 256 by 256, right? But what it is doing is that when we have the image, it automatically is extracting the images at different levels of zoom, capturing that effect.
Right. I'm just saying it can be made a tensor or you can also stack it.
Yes, yeah, absolutely. So the problem, so when I started working on this model, something that I was really passionate about is, can we build a model that is generalizable across different labs? And the problem that I faced was I set up a collaboration with 5 different clinics across the US. These were the images I got from lab 1. These were the images I got from lab 2. These were the images I got from lab 3. So the images are very different when you look at just the H&E stained images. So they are staining for the same marker. They are obtained using the same technology. They are just different. That is because of standardization errors. So I immediately thought that if I build a model on lab 1 data and I validate it from lab 1, it's going to give me very high accuracy. But now if I validate it on lab 3, it's probably going to fail on certain cases. So what I did was I developed a normalization strategy where for every single image, I created synthetically altered image. So these images, 4 images are not real. These are synthetic images, which would mimic certain contrast or variations that we would see across labs, and then train an algorithm that given any 4 images, it has to converge to this target image. So I call that neural network is NOMNet. And given these input images from 3 different clinics, it's able to normalize for that variation. And now that's what we use in our model development. So are all of you familiar with the confusion matrix? Okay, perfect. So I don't have to explain you what a confusion matrix is. So then we look at the confusion matrix, and this is when we split the training data. So this is the validation set coming from the training samples. And the confusion matrix is about 80 to 93% accurate across these diagnostic groups. And for the independent validation data, which was coming from 11 different patients, the confusion matrix is also similar. So you can see that the model is generalizable across these different data platforms. To show how this looks like for a clinician, because clinician is not concerned with a confusion matrix. They want to be able to see images. So here, when a clinician supplies an H&E image, the model is able to, in red was the bad color, right? In red, able to separate early stage tumor on the sample. And here on the second patient, the model says that there is no tumor present, all cells are normal. On the third, it's able to find the boundary that all the cells on the right here are early stage cancer and all the cells on the left here are actually normal, which was confirmed by a clinician. And similarly here, I'm just showing 4 representative images here, but as you know, there were about 9,000 of these images. So really the impact or the hope and goal of this work is that it can allow for rapid screening of images, especially in low resource settings where there are many patients, many images that are distributed across probably 1 or 2 clinicians. So this can help them to navigate these images, because what I think is that AI technology is still not there yet where it can operate standalone, because there is so much that you cannot really surpass the human pattern recognition, what is being happening and how do they explain it to the patients, but it can definitely act as assistive technologies, especially in low resource settings, both in terms of technology and in terms of expert knowledge, right? And it's translatable across pathology labs. And when we are able to develop methods, quantitative methods that are able to identify tumor early on, it can enable stratification of patients where we are actually intervening early on in the process rather than later on in the process. So I'm going to shift gears from here. So if anybody has questions on this part, I can take.
So, quick question. Would you say that on the one hand, time is a component, right? So we can figure out the negatives confidently, and we don't have to look at it no longer one set of systems.
Yeah, absolutely. So that's actually a huge problem. So imagine a clinician getting 300 slides to look at. And so there's this whole thing. If I give you 300 images, probably the first 5 images, you're going to tell me 10 things about it. But the moment you're on the 250th image, you're like, I'm done. These are the things. Okay, there's this red here, there's this green here. So there's this whole fatigue of looking at these images, which clinicians have to face, right? So can we screen those negative cases early on for them so that they can really focus on where their focus is absolutely needed, or where their expertise is needed? And also in terms of patients, when they have to wait for a negative diagnosis, that causes a lot of psychological stresses. So there are many biopsies actually that are done that do not have a positive diagnosis. So can we at least give that diagnosis early on? So it can help in both those two directions, according to me. Yeah. Okay. Yeah.
Sorry, I may have misunderstood this, but one of the things you pointed out was a lot of disagreement on how clinicians identify, right? So how are you sure that the data that's been annotated here is actually?
Yeah. Okay. Whenever I get this question, it's a proof that you were listening correctly. Yes. So that's a problem. But how do we solve that? Is if you look at the percentage disagreement are on the areas that are not very well defined with respect to their morphological characteristics, which we call as boundaries or interfaces. Whenever there are cells at the boundaries or interfaces, that's where the confusion is. But what we tell clinicians is that wherever you're confused, you don't mark that as your annotation. So we are not testing, or we are not taking annotations on any of those areas. We are taking annotations on only those areas where there is 100% confidence. And we're letting the model project on the remaining to see. Yeah. And the ultimate, so this also is not fully sustainable, because then how do you look at the accuracy of everything else that was not annotated, right? That's where annotations or ground truths generation from annotation would not be a measure, but the actual outcome of the patient. That's where we are closely working with clinicians to see what actually happened to the patient. When they did the surgery, did they actually find early stage cells, or did they find advanced stage cells? So that's where that would be the ultimate ground truth, which has no variability. Okay, any other questions? Okay. So now currently in my lab here at Udub Chemical Engineering, we are building upon some of these technologies, and also focusing on a little bit of new directions, which is how can we move towards personalized cancer care by using imaging and artificial intelligence. And the first area is, I talked about tumor, I talked about microenvironment, but one of the important cells in the microenvironment are the immune cells, okay? Immune cells are what are fighting the tumor, at least that's what we would believe. But what actually happens in the body is the cancer cells are able to trick some immune cells to thinking that there is actually a wound. What happens when we have a wound? We have all the resources of our body recruited to that place to make it heal faster. So some cancer cells are able to trick the system and make them believe that there's actually a wound and not something wrong going on. So all the nutrients immediately go to the cancer cells, and they're able to grow more and more. So there is this tug of war that is happening between the immune cells that got tricked and the immune cells that did not get tricked. So, and you can imagine wherever that balance will be, will change how a patient is going to behave with respect to that disease. And that actually explains why patients who have the same disease are getting the same treatment end up behaving so differently. So that's what we want to be able to identify and tackle in a much more quantitative and digital manner, to actually enable clinicians to get a much more personalized view of individual patients rather than one thing fits all. And also to kind of enable industries who are working in drug development to target some of these specific immune cells to help fight the cancer a little bit better. And the second is, how do we, using AI, integrate information that is coming from imaging and genomics to get more personalized indicators of what is happening with the cancer. And finally, something that I'm really passionate about is how do we take these technologies that are built only for one system, but take it to something that is going to be extendable to other countries and to other resources, especially developing countries where there are low resource settings, because it is believed that the molecular component of the tumor can be different from one community to the other. And unless and until we study those communities, we cannot really develop diagnostic approaches or treatment approaches that are suitable to those communities. So how do we bring in equity and diversity in this cancer research by using imaging and AI? So just to tell you why this is important, about a decade ago, breast cancer was categorized into these 4 different categories. And we don't need to go into the details of these categories, but these are the categories that are used today in clinics to identify which treatment a patient should get. But this was done a decade ago. And we have a lot of engineering innovation that is not currently being applied here. So how do we take this and go from finding immune subtypes between these subtypes that we knew from a decade ago that will advance our knowledge of how to deal with these patients? And then how do we connect that with imaging and genomic information? So in my lab, in addition to the research that I showed you on applying deep learning on clinical images, what we are doing is that we have this new imaging system where we can target multiple immune cells at the same time. So this is the image that we took in the lab. And each immune cell comes as a different color. And then we can use data science and artificial intelligence to look at the spatial correlations of these immune cells with respect to where cancer is and where the microenvironment changes are, and correlate that to the outcome of a patient. So there, the approach that we're following is staining the sample to actually image and scan it for multiple immune cells and annotate that and analyze it using supervised and unsupervised techniques to get a more comprehensive report for the patient. Yeah.
All the nutrients will flow there. Some will be tricked, some will identify which type of.
Yes, absolutely. So there are 2 ways to identify that. One is through some research, we already know which type of cells are actually tricked. So we know that these are the pathways that the cancer cells actually tricks. And the other is that we can actually do assays and experiments directly to see what pathways in the body is the cancer cell blocking for helping it to grow. And that's actually one way to find out new therapies. So we have immunotherapy that has come about where there is a checkpoint that actually suppresses our immune response. We all have that checkpoint and it's working. And why that is needed is so that we don't have a hyperactive immune response to everything that we are exposed to. But in cancer cells, because we know that they are blocking some immune cells anyways, there are drugs that can go and basically disrupt the function of that checkpoint. We inhibit that checkpoint. So your immune system has a total pass and it basically has a hyperactive response, which is needed, and it's able to cure or kill the tumor cells. But now it's only going to work in those patients where the cancer is growing based on that pathway, where it's actually taking cells and tricking them. So to be able to identify what are those patients is absolutely critical, because sometimes the side effects, like the benefit of this technology is really huge because you just needed to give it once. So how immune therapy works is that you take out immune cells from the body, you re-engineer them, basically make them more powerful and put them back in your own body. And now they grow automatically and they have that new ability, and that ability will stay with you forever. So it's a one-time treatment. So it's really powerful, but the problem is that if a side effect happens, it's going to be catastrophic. Why? Because you're playing with the most powerful part of your body, which is the immune cells, which have all the weapons and tools to really do whatever they're doing in your body, right? So the idea is that to understand this immune map and what's happening where is very important for us to take these new therapies into something that is more applicable to a broad patient group. Yeah. So just to show some preliminary data that we did is, here we took the H&E image, and just by applying unsupervised learning and a combination of supervised learning, we could identify where the immune cells were present. So here I can clearly see that the immune cells are present very close to the cancer cells in blue, which means that they are interacting. But let's say if there were not many immune cells here, but were far away from the cancer, that's when we know that the tumor doesn't have a lot of immune response. So will an immune therapy actually benefit that kind of patient? You don't know, because the tumor doesn't have a lot of immune response associated with it. And similarly, we can look at the changes in the microenvironment along with the immune cells, and with this, create a map of how immune cells are interacting with the tumor and the microenvironment to give a patient response. And I think I'm not going to go through this slide. But with this, I would like to thank you all for listening. And you can contact me through my email ID or our lab website. This is my lab. I started about a year ago, so it's a young lab. We have clinical collaborators without whom this work wouldn't be possible, because we are engineers at work, but we want our work to be inspired by clinicians and by patients, and all the funding sources as well as the department. Okay, and I'll be happy to take any questions.
Case where immune cells are immune cells are not tricked, right, by the cancer cells. Would there be a scenario, like, can you explain a scenario where the immune cells are not doing their function? Like, they're not tricked, but they're still not doing the function what they're supposed to do. Does that happen?
Yeah. So let's say an immune cell is supposed to kill the cancer cell, and it's not tricked. So its pathway is not blocked, but it's not able to reach the cancer cells because it's very far away, and there is not a lot of immune response that is elicited by that tumor, right? So how much immune response does our body elicit for that particular infection or disease or a foreign invasion is different, right? So if we don't have that many immune cells and they're very far away from the tumor, they won't be able to actually do that activity. Or sometimes when they don't trick the immune cells, they trick the pathway by which the immune cells are acting. Yeah.
So in that scenario, if we actually remove the checkpoint, then they will be able to reach it. But is there a risk there?
Yeah, so when you remove the checkpoint, obviously there's going to be more immune cells, but you don't know whether it's going to actually be enough to the point where it's going to be interacting with the cancer cells to that point.
So I understand that with breast cancer, things are localized. But what about things like lymphoma or cancers that are widespread? In that case, the issue of not being able to reach the cancer cells is not there.
Oh, no. So I think so the issue of not being able to reach the cancer cells is more of a signaling issue, and is not of a distance issue. So like if there is an immune cell through our lymphatic system, it's going to be present everywhere. So it's not like if a cancer is in the breast, then an immune cell is coming from the liver to kind of kill it. So it's not that. There are immune cells present locally within that tissue as well. But what is the pattern? Like if the cells are governed by very dense collagen fibers right next to them, and there is no immune cells within that vicinity, what is the migratory patterns of cells within that dense network of collagen? What is the signaling that is happening between the cancer cells? So like if you see, sometimes the cancer cells will change the properties of the collagen that is right next to them to change the signaling as well. So it's more of a signaling and migration issue rather than distance. Since you brought that point of lymphoma and leukemia, so for cancers that are a little bit less aggressive, like breast, the tumors sometimes elicit a little bit of a lower immune response. But for very aggressive cancers like leukemia and lymphoma, there is a very high immune response typically that we see. So that's where immunotherapy is actually even much more beneficial. And there are a lot of trials that are currently happening where patients have benefited from immunotherapy. It is just that we understand so little about the entire immune makeup that we're not able to extend that technology to all types of cancers and across different patients. I think we are doing it more from a perspective of the damage has already been done. Let's just use this technology to help the patients, and less from a perspective of, yes, we know all this, so this is going to benefit the patient.
Not the line one treatment, it's like second line.
Yeah, at least now. But I think now it's gaining a lot of new innovations, both from the engineering side as well as from the clinical side, because there was a Nobel Prize that was given in immunotherapy. So yeah, so now, and it is really actually benefiting a lot of patients, because when it benefits like the cancer is gone, right? So the benefit is huge. So the hope is that if we understand a little bit more about the immune makeup, we would not only be able to harness the benefit of existing immunotherapy technologies, but also kind of inform the development of new or combination immunotherapies that are going to work for patients. Because in breast cancer, there is a subtype, which is called a triple negative breast cancer, which is very aggressive, and it doesn't respond to any other therapy except chemotherapy. And chemotherapy has a lot of side effects. So they're having some of these other treatment line options for patients who do not respond to chemotherapy, or for patients where the disease comes back after the chemotherapy, can really be useful.
What's the relapse rate in breast cancer?
I'm not aware of the exact number, because I think that number changes for all of these subtypes and also the stage within that subtype. Yeah. Yeah.
You mentioned that there was 5 clinics, I believe, across the United States, and I'm not sure if it was the future research where you're going to check out the different demographic areas and medical areas. Do you think you'd be able to build another sort of model like you did with the NormNet to kind of generate the images that would look like sort of the genetic makeup that you see in the current set of data?
So the first part of the question, yes, that's what we are looking at currently, where we are going to sample patients from different demographics. But the second part, yes, we can use NormNet to kind of generate those synthetic images which are mimicking. But what we can really mimic at that point is just contrast, or just how the image looks like. But can we really mimic the genetic makeup by just inputting the H&E images in the beginning? So right now, the model has only learned from the images. It has not learned anything from the genetic makeup. So whether it can somehow normalize for that is, I don't think so. It will be able to, unless and until we put that as an input feature in the beginning and retrain for that aspect. Right? Because what you're seeing on an H&E image is just something that is morphological, right? And how much of a stain is it picking up? Whether it's darkly stained or lightly stained, and also an effect of the imaging. Like if I have scanner type 1, it's probably going to give you probably a bright image, or scanner type 2. Or if somebody who has, so like I have acquired about thousands and thousands of these images. So now if I take an image, it's less likely to be out of focus on any area. But if a new student takes an image, it's probably can be out of focus, can have some artifacts, right? So those are the kind of normalizations that model would be able to take care of. But the molecular normalization that you're talking about, that will have to be more intentional, in where we are parsing it through the feature space in the very beginning itself. Yeah.
I have a question with the first part of the lecture.
Yeah.
So you mentioned that infrared imaging is used. So firstly, what exact features do we get from infrared imaging? And secondly, would it be possible to directly use some kind of deep learning like CNN to get those features instead of
Mm-hmm. Okay. So these were the top features that, irrespective of what model I developed with my data set, just kept on coming as important and important in all my machine learning models. So basically, infrared is giving you the spectra, right? So we are looking at the height of every peak, and we are looking at the area of every peak, right? So those are our features. So when we looked at the height ratio of these two peaks, it gave us the information about the protein profile of the sample. When we took the area of this peak and ratioed it with the height, it basically gave us the information about the aliphatic chain length, or the extent of branching, which when you convert it to something biological, means that it's able to distinguish between different types of fibers, dense fibers versus loose fibers, because if you convert, so here we are basically converting chemistry to biology, right? So when you look at fibers that are densely packed, they're going to have a different level of branching in their chemical bonds that are present versus the loosely packed fibers. When we took the area ratio of these two peaks, it basically gave us the information about the DNA to RNA ratio that is going to change from something being normal or something being cancer. So these are basically the informations that we're able to capture using infrared. And whether we can extract something similar using deep learning on just the H&E image, well, it depends upon how many of these biochemical features are translating to morphology. And obviously, yes, we should be able to extract some of those features because the underlying hypothesis of the entire infrared work was that there is information in morphology that is correlated with the information in chemistry, right? But will we be able to get the exact information? I don't think so, because there's going to be some information that is leaking out when you're converting something that is biochemical to something that is completely morphological. I think you can still, the forward map of infrared to H&E is still easier because biochemical to morphology is still possible because the information about morphology is encoded in the biochemical aspect. But the backward map would be even more tricky because that's where you're taking from morphology to biochemical. Yeah, so it has a unit architecture. Yeah.
So it's kind of like a denoising.
I would not call it denoising. I would call it more similar to like template matching because I'm providing paired data, right?
Right.
Yeah.
Yeah. That's
yeah. Or like style transfer, basically. Yeah.
So, but even there, you kind of like, I'm curious about how you think this through four different synthetic things. Like you said based on contrast, right?
So we didn't pick that thing. So what we basically did was, yeah. So I'm just showing you 4 here, right? But I created millions of such pairs. So so basically this image has RGB, right? So we perturbed the red channel. It's data augmentation, right? And we perturbed the green channel and we perturbed the blue channel to the point where it gave us contrast, where some contrast was something that is clinically you will see. But in our training data, there were pairs where the synthetic image is something that you would never ever see in a clinical, like it's synthetic, right? But still, it was trained across that entire range to be able to learn this map.
And then you train across many images as well. So that's a huge.
Yes. We had to work on clusters to just train the deep learning algorithm.
Wow.
Because,
yeah. Days or
weeks.
Weeks.
Yeah, but if we had multi GPUs and if they did not break and had enough memory, right? Like, because these images can be large. So even if we had a very small mini batch size, sometimes they would just go out of memory, right? So we had to have multiple GPUs that we were accessing at the same point. And that would even take days. So we used to save after every epoch just to salvage whatever we can to start seeing if the model is going in the right direction or do we need to just break it and do something else.
Yeah. It's nice to see this because we were talking about data augmentation in class as well.
Yeah.
And using encoder, decoder, mostly auto encoder. But here it's kind of very similar. Just keep the inputs.
Yeah. So
it's nice to see the application of that.
Yeah. Here the data augmentation is happening from a very different perspective. Yeah, where the data augmentation is not happening from a perspective of training the algorithm per se, but to generate data to. Yeah, exactly. Yeah, because it wasn't possible really to get a lot of images from only one clinic or multiple clinics to mimic what that lower or upper bound of variations can be. Like I saw these variations, but whether these are the lower or upper bounds, there is no way to know.
It's good. It's interesting that you need to train with so many variations in the inputs that you're providing.
Yeah.
It's actually able to learn something on how to normalize problems.
Yeah.
You said like when a new student would be taking this picture, how much would a blurry image affect the results? It can be pretty fine grained, but that you need that like really fine grained aspect to affect the actual.
Yeah. So basically, because right now in our model, okay, let me go back. Yeah, right now in our model, the images are coming at different levels of zoom, right? So it's coming at 5x zoom, 10x, 20x, and 40x. So because all of them are mixed together, there is no way for us to know whether a 20x is absolutely crucial or not. So if, let's say, most of the discrimination is happening at the 5x and 10x level, so if a 40x image is slightly blurry, it would still work. Right? So that is something that we don't have the answer to, and even the field doesn't have the answer to, as to what zoom do you absolutely need. And probably that is something that we should find out by just supplying images of different levels of zooms. But with this, we cannot say that conclusively of how much blurriness would actually affect this model. If there is a lot, yes, it would, but how much of it would be, and whether it's systematic or not. If it's randomly distributed, then as long as we have enough training data, the model should be able to account for it, because at that point, then it would just act as data augmentation. But if it is systematically distributed only across certain types of cells that you're discriminating against, then it would be a problem, right? Then it is something that you need to kind of account for in the training. Yeah.
I will notice that on the lower left corner, it's patent 2020. Is this method being patented?
Yeah, so we basically patented the pipeline of being able to, for the first time, show normalization working across different labs, showing a deep learning that is translatable across different labs, and detecting tumors early on, which is where we are focusing on segmenting early stage tumor. So that's the patent. Yeah. Yeah, it's a good question. So for people to use it right now, there are collaborators that we have that use some of our algorithms to give us feedback. But for it to be used in a pipeline where they're using it as a tool to sign off a patient report, this is right now a technology that we have developed, not approved by FDA, not approved by any technology or any, I would say, agency to make it actually something that is part of their diagnostic pipeline. Because then there are issues of how, like, what can be the percentage inaccuracy, how is that affecting? Like, what are the is compliance issues? Like, how does this algorithm input the data? Can you put that on a cloud platform? If you put it on the cloud platform, how can you put patient data on the cloud platform? Will it need metadata or not? Right? Those are the things that you need to do, and then get it approved by an agency to actually become a part of a diagnostic test. So right now, this is a model. It's not a diagnostic test. Right? So that journey we haven't taken yet, but our clinicians, they're using it to give us feedback to see if it's useful or not. But the thing is, for us to be able to do something where we file it for the FDA or to get it approved in that space, we have to really work on every single part of that pipeline. And that's where sometimes from research, you see a lot of startups, like graduate students or postdocs would go open a startup and actually work just on that, because that's a whole area on its own where you're focusing on translating something to being a diagnostic test. Yeah, and I haven't started that startup. Maybe a student of mine will, so we'll see. Yeah. Yeah.
I have a question regarding the scientific information stated in our science. So the paper which you published regarding it, the test data set, that also has specific data set, or was it the original samples from the last?
So are you talking about the previous paper?
Yes.
So this paper is not yet published yet. So that's the thing. So the previous paper where we worked on the inertia indicator, there we didn't do synthetic data generation. Yeah, but how we basically did the synthetic data generation is by just perturbing the color channels of an image.
So the test data